Thank you very much. Just a few info. So Professor Mansvelder actually received his PhD in neurophysiology in Amsterdam at the Vrije University, studying calcium channels control of vesicle release in neuroendocrine pituitary cells. Then he moved to the US for 2 postdocs. During his first postdoc, he studied drug-induced synaptic plasticity in dopamine neurons and uncovered how nicotine receptor modulate synaptic transmission in the VTA. He then moved to a second postdoc at the Columbia University, where he examined rapid calcium dynamics in dendritic spines, mainly using 2-photon imaging. He then came back to Europe and to the Netherlands in 2002, where he started his own lab at the VU University in Amsterdam, and then is there since 2002, and he became full professor in 2008, and also heads of the Department of Integrative Neurophysiology, SISSA. His current research focuses on how the prefrontal cortex and subcortical brain areas orchestrate attention behavior in rodents. And in addition, his lab also investigates anatomical and functional organization of human neocortical microcircuits and how properties of human neurons relate to human cognition. So thank you very much for being here today, and we're looking forward to listen to your talk.
Thank you very much for the kind invitation to come to Trieste and tell you about the things that we do in Amsterdam. I probably share with many of you the fascination for the brain, but the human brain in particular. We study, of course, as was mentioned already, how neurons give rise to cognitive behavior and use rodent research to do that. But of course, it's most fascinating to see how in the human brain neurons give rise to human specialized cognitive behavior. And today's talk will be focused on that. I will have 3 parts. I'm not sure if I can talk about them all, but we will see how we will do with time. First part is a little bit longer, and then 2 other parts, and they cover, let's say, a lot of levels of organization, so hopefully there's something interesting for all of you. So, as I said, of course you can use laboratory animals and other animals to study how the brain and neurons in the brain organize cognitive behavior. And because, of course, other animals also show a lot of cognitive behavior, and to talk about our closest relative, chimpanzees, of course, they can show amazing behaviors, use tools to get food, and when you train them in the laboratory, they can even do amazing working memory tasks. I don't know if you've ever seen movies of Ayumu, is his name. He can memorize the location of 9 digits in order in a short amount of time. You should watch on YouTube because the numbers are flashed, let's say, half a second or less, and then he can immediately point where 1, 2, 3, 4, 5, 6, 7, 8, 9 are without them being there, of course. If you try this yourself, I'm sure that you will have a hard time. I had a hard time, at least, doing that, getting up to 7 at least. So it's really amazing. So, but of course, when you compare that kind of cognitive behavior, compare that to human behaviors with arts and sciences, there's a big gap, and it fascinates me. I'm sure you're also wondering what is different about the human brain that it can explain, that it can account for this gap in cognitive ability, mental ability. Well, of course, one of the obvious, what from neurobiology, so the questions of the talk will be, what are the neurobiological factors that drive human mental ability? And of course, we're gonna look at neurons, and we're gonna look at the brain and cortical architecture. But the most important thing, of course, is we can speculate about what these neurobiological factors might be that drive mental ability. We do that all the time. It's much more fun if you have a glass of wine with that. But can you actually test whether these factors are actually contributing to human behavior? So that's what I'm going to show you. Also, our, let's say, initial attempts to really try to put this to the test and see if we can actually get data on that. Now, obviously, the first factor is humans have a much bigger brain than most other animals that we study in the laboratory. So it's no wonder that with such an extended cortex, neocortex, and other parts of the brain, that the behavior that's generated by those circuits can also be much more elaborate. And also when you look at the amount of neurons in the cortex, you see that humans, they have many more neurons in the cerebral cortex, 19 billion of neurons in the neocortex compared to other animals with big brains. That's a lot, but there are whales, this pilot whale, for instance, that has even more numbers in the neocortex. So there are animals with big brains, with a lot of neurons in the cortex that don't show necessarily human-level cognitive ability. Although, of course, I have to admit I don't know what the whale is thinking about, but maybe it's also all kinds of mathematics that I have no clue about. But let's say, I don't think that a big brain by itself can explain all that gap between that can explain human cognition. And the same holds true for the number of neurons in the cortex. Even a lot of neurons in the cortex does not necessarily mean you get human cognitive abilities. So what are other factors? So also microarchitecture of the cortex is different in human cortex. So with the cortical expansion, evolutionary expansion, not only the surface area of the cortex increased, but also the thickness of the cortex increased when you compare to other species. And when you put this at an absolute scale, you see that the layer 2, 3 is in particular large, but also when you put this in a relative scale, then you see that the human cortex generally contains much more layer 2, 3 than the other layers when you compare it to the other species, primates, other carnivores like cats and dogs, and rodents. So the question is, do any of these parameters, do they contribute to human cognition? Is there any relevance for human cognition? So we wanted to put that to the test, and the way we went about that is we started a collaboration with neurosurgeons in Amsterdam. They operate on different groups of patients on a weekly basis, epilepsy patients, tumor patients, and so for epilepsy patients, usually the source of the epilepsy is located in the hippocampus, so inside the temporal lobe. And on their way to, so when the surgeon decides, well, okay, this patient is pharmaco-resistant, so we have the ability to remove the part of the hippocampus, so then treat the epileptic seizures, and then of course the surgeon has to get into the brain to cut out the hippocampus. On the way to the hippocampus, there's a lot of other tissue, cortex tissue taken out, which is not necessarily part of the disease, but which we can study in the lab and see if there's properties in those microcircuits that relate how these properties compare to, for instance, things that we know from rodent brain. So the medial temporal gyrus, of course, we get the reason why we get a lot of this tissue is because, of course, the hippocampus. But what is the function of this area? So what people have observed is that when you look at the properties of the temporal lobe, that they correlate with all kinds of aspects of intelligence tests, IQ tests. And so, for instance, the thickness of the temporal cortex, here area 21, correlates positively with IQ scores of healthy subjects. But also in the same papers, they show that when you look at the activation of these cortical areas, then the amount of activation correlates with IQ scores in healthy subjects. So we wanted to know, well, of course, if we get a lot of tissue, if we get tissue from that area, do the properties of this tissue also correlate with IQ scores in the patients? So you have to know that when a patient undergoes brain surgery, especially an epilepsy patient, before they will have the surgery, the days before, they will go through a whole series of tests, cognitive tests. So the medical psychologist wants to find out, well, what's the baseline psychological level that you function at before the surgery? then the surgeon is going to take out tissue, and then when they come back to the psychologist later on, he's going to test, or she's going to test again what is your level of functioning and what has been affected by the surgery, so that we can design specific therapies to improve that. So there's a whole lot of data on the same patient that we get tissue from on their cognitive performance. So we made use of that data, and also, of course, there are MRIs of the same patients. So Natalia looked in the lab at the thickness of the cortex in the patients and correlated the thickness of the temporal cortex with the IQ scores of the same subjects in the patients, and found that there's a similar 0.3 correlation that was described in the literature for healthy subjects. Correlation between IQ scores and cortical thickness. So people with higher IQ scores tend to have a thicker temporal cortex. Now, do all the layers contribute to this association, all the layers in the cortex? So what we did is we took the tissue of the patient and divided that in the different layers and different sections across the crown of the medial temporal gyrus, and started quantifying the size of the different layers. And of course, this example already gives it away a little bit, because when we correlated the thickness of layer 2, 3 to the IQ scores, then there was a very strong correlation with patients with high IQ scores. They have a much thicker layer 2, 3 than patients with low IQ scores. And not only that, when you look at the microarchitecture inside their cortex, then you can already appreciate from these examples that the density of neurons is very different in the two examples. So thicker cortex usually has a much lower density of neurons than thinner layer 2, 3. And indeed, when we quantify that, you see that the thickness of layers 2, 3 show a negative correlation with the density of the neurons in that layer, and also there's a positive correlation between the thickness of layer 2, 3 and the size of the neurons, so the cell body area of each of these neurons. So these patients with a thicker layer 2, 3 tend to have neurons with larger cell bodies in layer 2, 3. So, of course, the question is, well, what type of cells are these? Are they pyramidal cells? Are they interneurons? And what type of them are they? So what we do in the lab in collaboration with the Allen Institute is we patch neurons in the tissue that we get from the surgeon. We keep it alive. We put it under the microscope and patch the neurons, fill it with a dye that's called biocytin to later on reconstruct the morphology of the neurons. We record the physiological profiles of these neurons, and we take out the nucleus after the recording and do a complete transcriptomic profile, so an RNA expression profile of that same neuron. So we get 3 data modalities: molecular data, physiological data, and morphological data of the same neurons. So when we do that for all the neurons in the pyramidal neurons in layer 2, 3, we find that there is a whole range of neuron types based on their transcriptomic profile. So when you group them based on their gene expression levels, you get 5 big groups, and that are located throughout layer 2, 3. So you have superficial ones. They are very much related to the mouse pyramidal neurons in layer 2, 3. And then you have deeper ones that are quite distinct from the ones that you find in the mice. So we also, of course, reconstruct the morphology of these neurons, and we have the physiological profiles of these neurons, and especially these deeper, larger types of pyramidal neurons. They are very specialized in the human cortex that you don't find in the mouse cortex, for instance, and they are also the first ones to go in Alzheimer's disease, we found in the literature. So these are neurons that are very prominent, let's say, candidates to support human cognitive functions. So these are the big neurons, the ones that we study. And the next question is, does the size of the neurons relate to cognitive abilities? So of course, when you have a thicker cortex, as we already saw, the thicker of the cortex is in patients with higher IQ scores, the bigger the neurons are that can be in there. So they have larger cell bodies. So, but are their dendrites also larger? So indeed, when we reconstructed the neurons, their biositin fields for each of the patients, then we see that patients with thicker cortex also tend to have larger pyramidal neurons with larger total dendritic length. And also the total dendritic length correlates with the IQ scores of the patients. So the patients with higher IQ scores tend to have larger pyramidal neurons in their layer 2, 3, deep and layer 3. Also, when we just look at where we could do, when we could not reconstruct the entire morphology, we just took the diameter of the cell body, and that also correlates. Or when you separate high IQ scores and low IQ score patients, then you see that the cell body size also is larger in the high IQ group. So these cortical microarchitectural features, they correlate with IQ scores, and they set human pyramidal neurons in layer 2, 3 apart from, for instance, mouse pyramidal neurons. So the total dendritic length is about 3 times larger in human pyramidal neurons than in mouse pyramidal neurons. Our colleague in Madrid already had described that when you look at the number of synapses that pyramidal neurons in layer 2, 3 receive, that in the human cortex, neurons receive about twice as many, or more than 30,000 synapses per neuron, whereas in rodents, this number is about between 15 and 20,000, so much lower. And so these neurons, they receive, they not only have much larger dendrites to receive more synapses, but they actually receive more synapses when looked under the EM. So the question now is, what are the properties of these synapses? Do these excitatory synapses also show functional specializations when you compare them to, for instance, rodent synapses? So to test that, what we did is we made paired recordings. So we recorded from pre- and postsynaptic pyramidal neurons at the same time in both human cortex and in mouse cortex, and stimulated the presynaptic neuron with an action potential, recorded the postsynaptic response, glutamatergic inward current, and compared that between mouse and humans. What we observed is that, in particular, the amplitude of this EPC is much larger, so of these synaptic currents is much larger in human pyramidal neurons compared to mouse, but also the failure rate is much lower. So what is failure rate? So when you record this kind of synaptic transmission in the mouse cortex, what you see is that each action potential does not necessarily result in a postsynaptic response. The synapse also fails 1 out of 4 times. It does not show a postsynaptic response. So the failure rate is about 25% in the mouse synapse, where in the human synapse there's almost always a postsynaptic response. And our colleagues in Hungary, from Gábor Tamás' lab, already showed that excitatory synapses in the human cortex, they have a much larger size presynaptic bouton. So what you look here at is an EM micrograph of a synaptic structure. You look here at the synaptic cleft. Here you see the presynaptic vesicles. They are docked to be released in the glutamatergic synapse, and this is in the rat, the same structure. And what he observed is that in the human synapse there are more vesicles docked, ready to be released, than in the rodent synapse. And it means that when an action potential arrives, there's just more vesicles available for release. So that's why we think the reliability, the amplitude and the reliability of these synapses in the human cortex is larger. So what we also observed is that in these paired recordings, when we looked at short-term plasticity, so when you stimulate the presynaptic neuron with a train of action potentials and record the postsynaptic response, you see that there's a depression of the response. So the first amplitude is much larger than the last in the train. And the same holds true in the mouse, so you just need to decompose this a little bit. But when you then look at the recovery from depression, so we wait half a second, and then you see that the human synapse is already back to the amplitude at the start of the train. But the mouse synapse still has the same amplitude as the last in the train. So there's no recovery, or very slow recovery from depression. When we quantify that and calculate the time constant of recovery, then you see that in human cortex, this is about 150 milliseconds. whereas in the young mouse or in the adult mouse, these time constants are about 3 times longer. If you would do this experiment, this experiment has also been done, for instance, in ferrets, then it's even 1 second the time constant of recovery from depression. So this is, you might think, well, this is a very detailed observation. Does it actually matter? So we collaborated with Michele and Juliana here in the audience, and he used the formalism that was based on Shannon's information transfer between 2 telegraph stations that has been worked around and be applied to synaptic transmission by Misha Tsodik's and Idan Segev's groups, and to use this mathematics to calculate what is the information transfer in synapses with fast time constants of recovery versus synapses with slow time constants of recovery. And what he observed is that when there's slow recovery from depression, then when the presynaptic neuron is increasing its firing rate, the amount of information that's transferred does not increase much anymore because the synapse depresses and cannot relay more information. But when the neuron recovers from depression fast, and then you see that much more information is relayed with increasing firing rate. So this little, let's say, tweaking of this parameter in the human synapse matters a lot for how much information neurons can transmit. 3 times, well, much more information, especially when you look at high firing rates. So to sum this part a little bit up, the human neurons, they have much longer and more complex dendrites that receive more synapses, that are stronger and more reliable, and that recover faster from depression and thereby can transfer more information. Well, but the big point, of course, is it's fine that you can transfer more information, but when a neuron receives all these synapses that are stronger and more reliable and transfer more information, can the neuron actually do something with that information? Can it encode that somehow in its output? So what we did is, so the question is now can human pyramidal neurons encode synaptic inputs into extrapotential output? And how well can they do that? So what we did is we, also inspired by experiments that Michele did in the past, we were hiding known frequencies of synaptic inputs in the recordings. Those frequencies would not generate extrapotentials by themselves, but the question is, do those frequencies, do they matter for the extrapotential output? Can the neuron time its extrapotential output based on those hidden frequencies? And to make a long story short, so the neuron fires, we depolarized the neuron a little bit to make it fire at 10 hertz, and then we applied known frequencies that were hidden in the noise, and we then quantified the extrapotential timing relative to that hidden frequency. And you see that at 10 hertz, the extrapotentials, so the firing rate, is higher at one part of the sine wave than at the other part of the sine wave. So although the sine wave did not induce extrapotential firing, it did skew the timing towards a certain phase of the frequency. At 100 hertz, this still was the case. It's a little bit shifted, but there's a preference of firing of the neuron at some part of the 100 hertz phase, so that indicating that the neuron is sensing this low amplitude input. And even at 1,000 hertz, we still observe that, so the neuron is not firing at 1,000 hertz. The neuron maintains its firing at 10 hertz, but when it fires, it is related to that 1,000 hertz sine wave. So that means that the neuron can really encode these kind of frequencies in its extrapotential output. So when you put that in a curve, then it looks like this. You see that for the human neuron, up to 1,000 hertz, it can lock its extrapotential output to the synaptic input, to that frequency. When you would do the same experiment in rodents, then you see that already at 200 hertz, the frequency starts to drop off, or the phase locking starts to drop off, and the rodent neuron cannot follow up to 1,000 hertz, but human neurons can. So they can encode much more frequencies, much more, but they have a much larger bandwidth to encode in their extrapotential output. So what is the mechanism underlying this? So what is necessary for a neuron to be able to respond to fast synaptic input with a high frequency content? That's a fast action potential generation mechanism. So as long as the neuron it has a fast electrical mechanism to respond to synaptic output, then it can encode those kind of frequencies in its action potential timing. So we looked at the action potentials of human neurons and of mouse neurons, and at different frequencies, firing frequencies. And what you observe is that in the human action potentials, the frequencies do not change the waveform much. Here you look at the derivative of the action potential. You see that it is very similar in shape for the different frequencies. Whereas the mouse neuron, the shape of the action potential starts to change much more with increasing frequency. So you see that the membrane potential change starts to slow down at higher firing frequencies. That means that when the mouse neuron is firing at 15 Hertz, that the action potential becomes much slower and will not be able to encode fast inputs into action potential timing. So we think that this fast action potential mechanism can underlie this wider bandwidth of encoding input into output. Now I gave you a lot of detailed information, but of course, why would you care? What does it matter? I mean, does this rise speed of the action potential is super fast, microsecond scale? Does it actually make a difference for human cognition? So again, as we started out, can you actually test this? So since we had IQ data of the patients and action potential data of the patients, Natalia compared action potential kinetics, rise time kinetics, in patients with high IQ scores and with low IQ scores. And what she observed was that the action potential in patients with high IQ scores does not change much of its waveform with increasing frequencies. But in lower IQ patients, the action potential changed much more in its shape, the rise speed. You see that quantified here. And when you look at the correlation, you see that with increasing IQ scores, the action potential rise speed remains fast at this kind of frequencies. So we think that this kind of action potential stability, especially when neurons start to fire more, for instance, so temporal cortex is involved in language, and people have recorded in vivo in patients action potential firing during, for instance, reading a book or memorizing words. Then neurons can fire at 20, 30 Hertz. So these frequencies are relevant, and especially when people are concentrating on texts, for instance, the neurons can fire repeatedly, and we think that this maintaining a fast action potential allows neurons to maintain a very high bandwidth input-output encoding capability. And when they slow down more, so in people with low IQ scores or in mice, that this encoding is much reduced by slowing down of the action potential at high firing rates. So it is relevant for cognition, but what are the underlying mechanisms? So to test that, we recorded sodium and potassium currents in neurons from humans and mice, and applied action potential waveforms to see what the stability was of these currents. And what we observed is that during action potential waveforms, the human sodium current already looks a little bit different than the mouse sodium current, but especially with repeated action potentials, you see that the human sodium current, the amplitude reduces less than the mouse sodium current. The amplitude reduces more, and also the potassium current reduces more in mice than in humans. And this is the quantification. So even at the second action potential in humans, you already, the mice already lose about 50% of the amplitude of the sodium current, whereas in humans it's 20%. So what are the properties, the voltage dependence and current kinetics properties of these currents? So I'll go over this quickly because it's maybe too detailed, but anyway, what you can observe is that the sensitivity of these sodium channels to the membrane potential is right shifted both for the inactivation and the activation of those sodium channels. And also the inactivation time constant, so the rate of inactivation is much slower in human than in mice, but in particular the recovery from inactivation is faster in humans than in mice. So that means that the human sodium currents go less into, need a higher voltage to go into inactivation. They go into inactivation faster, slower, the humans go slower, and they recover from inactivation faster. So what are the consequences of that? Probably means that the availability of sodium channels for the next action potential remains higher. So to test that, we wanted to know which properties of these sodium channels can explain the differences we observed in, for instance, synaptic encoding into action potential timing. To do that, we used computational modeling. So we made a model of these sodium channels either with complete mouse properties or with human properties. And then we systematically varied individual aspects of these channel properties from mouse to human. So when we did that for the activation properties, we saw that the sodium current properties remained the same as in the mice. But when we started to include human inactivation properties, then the sodium current started to get these human sodium current properties. And also when we looked at the same experiment that I already described, so the encoding of synaptic input into action potential output, that is affected by these properties. So when you give the channels human inactivation type properties, then the bandwidth of encoding is larger than when they have only mice properties. Okay, so there's a molecular reason why human neurons can encode synaptic inputs into action potential timing. It's because their sodium channels are remain much more available for the next action potential. They show less inactivation. So to sum that all up, so we see that the structure of the cortex and the properties of neurons associate with cognitive ability in humans. The neurons have a large synaptic information transfer due to several properties related to synapses and recovery from depression and related to action potential generation mechanisms. Sodium channel function enables stable action potential kinetics that provide a larger bandwidth for encoding. So we think that these kind of properties are really helping neuronal input-output function that is relevant for human cognition, both the structure and the function. So I think we have time for a second part. So one of the things that is observed, of course, across many species, I'm going to switch gears a little bit, across many species is that when you look at the different oscillation frequencies that you can record on the outside of the brain, for instance in EEG, is that when you look at the different brain sizes, that the frequencies of these oscillations is very similar in the different bands. So when you look at the gamma band, you see that it is always around whatever 40 hertz, irrespective of the size of the brain. So in mouse brain, rat brains, cats, macaques, humans, it has a very similar frequency. Only the theta frequency is brain size dependent. So of course at the same time when you compare these different brains, the small one on one end and the big one on the other end, is that there's very much differences. There are many differences in the microarchitecture of these cortex. You see that the density of the neurons in the mouse cortex is much larger than the density of neurons in the human cortex. And it makes you wonder, so if this electrical signaling, these frequencies, fast frequencies, slow frequencies, are all very similar, how come that neurons that are bigger, that are further apart from each other, how can they maintain these kind of frequencies? Or to put it in a different context, so when you look at a microcircuit from a pyramidal neuron to an interneuron to a pyramidal neuron in these different cortex, how can it be that the time that this microcircuit operates at is very similar in generating enables them to generate these rhythms with equal frequencies? How is it organized? These neurons are much more larger and the distances are much larger, so you expect also bigger delays and stuff like that. So we looked into that because we've, together with the Allen Institute, we've recorded from many interneurons in the human cortex throughout the layers, the different types that you all have heard of probably, so the VIPs and the parvalbumin ones and the somatostatin expressing interneurons. You can also find them back in the human cortex. And we used, of course, the transcriptomic profiling as well, morphological profiling, as well as physiological profiling in these neurons as well. So what we did, we asked the question, well, if you look at two of these, for instance, parvalbumin neurons, and you compare the mouse parvalbumin neurons to the human parvalbumin neurons, they are structurally very different, but physiologically they also have some differences. So how can we explain this timing in the microcircuits that underlie these oscillations in for instance, gamma oscillations? So what we did is we looked at first we looked at the morphology of these parvalbumin neurons, quantified the dendritic morphologies. And what we observed is that in parvalbumin neurons in humans, the total dendritic length is a little bit longer. The number of branch points, however, is much smaller, and also the number of dendrites that come out of the cell body is lower in human parvalbumin neurons compared to mouse parvalbumin neurons, fast-spiking neurons. So, is that relevant? How does this affect the synaptic propagation in the neuron? So we first used a model, a computational model of fast-spiking neurons to see how the location of the synapse on these different dendrites that have the properties of either human or mouse parvalbumin neurons, how that affects synaptic transmission. And in the model we observe systematically that when you have a synapse on a human morphology, you always have a lower amplitude. No matter how far you are from the cell body, you always have a much lower amplitude in the human synapse compared to the mouse morphology, synapse on the mouse morphology. So do we actually observe that in the cortex? So when the synaptic potential smaller in human fast-spiking interneurons. So we made paired recordings, and between pyramidal neurons and fast-spiking interneurons, and what we observed is actually that these synapses have very similar amplitudes. So when we quantify that, you see that the amplitude in human and mouse fast-spiking neurons is very similar, and also the onset latency, so how fast can you record that postsynaptic response after the action potential is very similar in those two species. So what can explain the differences between the prediction from the model and the experiments? So we used again the model to systematically go through different possibilities. So we changed the human morphology step by step into a mouse morphology and the other way around, and then looked what was the effect on the EPSP amplitude in the model. So when you take a full human morphology, then we saw already that you get a very low synaptic amplitude. When you change the path length to the mouse path length, so when you make the synapse closer to the cell body, let's say, then the amplitude increases. But when you change the dendrite count for a few dendrites from the soma to many dendrites from the soma, as is the case in the mouse neuron, then the amplitude drops again rapidly. And the other way around, when you look at the, also this I already said, so when you look at the mouse morphology, they change the path length to the human morphology, then you see a drop in the amplitude, but when you then reduce the number of dendrites that come out of the cell body, then suddenly the synapse amplitude goes up. So at the same time, when you increase synapse strength on the dendrite, so you make the synapse twice as strong, that helps in the human morphology, that helps the synapse a lot in its amplitude. So we think, and this reminds us again of the same paper that I already showed, that human excitatory synapses are larger than mouse and rat synapses. So we think that both the longer dendrites of the humans are compensated by fewer branches that come out of the cell body and thereby increase the amplitude, and also stronger synapses on the dendrite. So then the next question is so this is the input to the fast-spiking interneuron, right? So we think that there are structural and functional adaptations, but is the output of the fast-spiking neuron different? So we recorded from the fast-spiking neurons and then looked at the action potential, waveform and properties. And what we find basically is that the human action potentials are a little bit faster. Write speed is faster, and also the fall speed is faster, so the human neurons generate action potentials a little bit faster. And to test what happens in the models, even with the longer dendrites, you get faster action potential initiation in the human morphologies. So with increasing size of the neurons, you see that the axonasal segment starts to fire earlier compared to the cell body than in the case of the smaller neuron. So can fast-spiking interneurons generate fast synaptic response in the pyramidal neurons? So we made paired recordings between fast-spiking neurons and human pyramidal neurons and mouse pyramidal neurons. And what we observe is that, this is the experiment, so you record from the presynaptic fast-spiking neuron, record in the postsynaptic pyramidal neuron, and record the inhibitory input coming from the fast-spiking neuron. And what we observe is that when you quantify the onset latency, so the time it takes to between the action potential of the fast-spiking neuron and the synaptic response in the postsynaptic pyramidal neuron, then we see that there's a population of human synapses that is very fast. It's much faster than the mouse synapses. And when we quantify that, you see that the onset latency in human neurons is a little bit faster and the amplitude is a little bit larger. So when we then collapse all this information into the timing in this loop that I started off with, going from the pyramidal neuron to the fast-spiking interneuron back to the next pyramidal neuron, that is the mechanism that generates, for instance, gamma oscillations in the brain, then you see that there's several adaptations that compensate for the differences in distances between the neurons and the sizes of the neurons. So so when you then overall calculate all this time it takes of these different steps, then you see that the overall time is very similar between the two species. You see that the human fast-spiking dendrites are longer, but the synapses are stronger. The initiation kinetics is faster for the action potential, and the pyramidal neural inhibitory connections are a bit faster, and thereby the timing of that loop is very similar. So so this is the second part of the talk. In view of time, I will only go over the third part in a very short amount of time. I will skip a couple of slides, but I still think it's an exciting part because it deals with the transcriptomic part of the data. So when you look at the literature of the neurobiology of cognitive ability in humans, then it is split in two very far apart fields. On one side, you have the brain imaging that I already showed some data from, that the thickness of the cortex in people with high IQ scores correlates with IQ scores in certain parts of the brain. And on the other side, you have the genetics research. So the genome-wide association studies use large cohorts of humans to correlate SNP, so single nucleotide polymorphisms, to IQ scores, and they find a lot of genes. So here there's a lot of brain areas that are associated with intelligence, and on the other side there's a lot of genes that are associated whose polymorphisms are associated with IQ scores. So of course, the most interesting thing is how do these gene effects express in neurons, how do they generate the neuronal properties and the brain structures that lead to the observations that are done in the brain imaging. So we, of course, find also some correlations of these neuron properties and structural properties and physiological properties with IQ scores. So in the study that I'm now going to very quickly summarize in 2 minutes, what we wanted to know is, can we use the transcriptomic data, because we have gene expression data, so which genes of these intelligence genes are expressed in pyramidal neurons that potentially could give rise to these kind of differences in brain imaging related to intelligence scores. So we used the transcriptomic data that I already explained, looked at the gene expression profile in the different neuron types that we identified, and we've asked the question, does expression of genes of intelligence correlate with neuronal properties? So to make a long story short, we look at correlations of gene expression in, for instance, dendrite size. And what we find is that there are, so when you use different IQ data sets from different papers, IQ or educational attainment, which is a proxy for intelligence in humans, and relate them to human accelerated regions, which are regions in the genome that are especially show especially fast mutations only in humans relative to gorillas and chimpanzees. Then what we observe is when we limit this data set to the genes that are really expressed in the neurons that we record from and that are really correlated their expression with, for instance, the size of the neurons and the speed of the action potentials, that there's a much larger proportion of these intelligence genes that are related to human accelerated regions that are located in regions that are specific for human evolution. So what are these genes? So they are mainly synaptic genes, a lot of synapse organization, glutamatergic synapse, synapse membrane, and are when you do a gene ontology analysis, you find them back, and many of the synapse-related genes are expressed in neurons that are whose shape and function are correlated with intelligence. Okay, so to sum this talk up, or to end this talk with a lot of speculation, what we've seen is that brain expansion and evolution of mental ability relies, of course, on a bigger brain. Of course, humans have a much bigger brain, so there's an increase of brain per body mass that definitely has a big impact. Expansion of frontal areas, so areas that are related to cognition, is most likely contributing. And there are more neurons in the brain and in the cortex. That helps a lot too, of course. But they scale linearly with this ability. But if you look at the neuronal properties, faster, large and complex dendrites, more synapses per neuron, per synapse more information transfer, and faster action potentials. All these properties hold true for every neuron in the circuitry. So multiply these properties by a very large number of whatever 100 billion neurons in the brain and even more synapses. So you get a very steep, let's say, potentially, if this would happen to all your neurons and synapses, that would create a lot of increase in computational power of the brain. So what I think is, just to put it forward, is that there's of course a lot of influence of brain anatomy on how mental ability is supported by our brain. But I think that as soon as you take into account synapse and cell function and molecular function, then of course you can really start to see why there's such a big gap. So there's all kinds of specialization at the molecular and cellular level in the human brain that we cannot appreciate fully yet because it's so hard to record from these or to get to these neurons, that can really explain why there is such a big gap in cognition between the different species around us. Of course, it's all speculation, but at least you can take it home to think about it a little bit. All right, there's a lot of people I need to thank that did the work in the lab. Of course, Natalia and Christian for the morphologies and the PhD students. Our collaborators, of course, in Hungary collaborators, in Jerusalem the collaborators, in Italy nowadays. Back then he was much closer by in Antwerp, but now anyway. Still great to come to Italy. And our collaborators at the Allen Institute. And the funders, of course, and I would like to thank you for your attention.
Thank you very much. Do you have questions? Okay, start here.
Thank you for the very nice talk. I have questions, curiosity. The first relates to these properties that you described of layer 2 and 3, and I was wondering whether these properties you observed only in, you had a chance to observe it only in temporal lobes, or you just also explored it elsewhere? So
Right.
that's the first question.
Mhm.
So second is, why L2 and 3? So why those layers? And
Mhm.
if, I guess it's for the horizontal connections maybe. So also the question is, okay, have you looked at neighboring? So are neighboring also structures have exactly similar properties? And third is, IQ is a very global measure of intelligence, right? And you are exploring a very specific piece of the brain, which is temporal lobe, which has functions that are well known, like memory. So I was wondering whether you also try to explore some of the subtasks that go into the IQ, like memory performance, if you just try to also look at some specific abilities of that goes into IQ. Thank you.
Excellent questions, of course. So the first one was the region-specific, only temporal lobe or frontal or others. So we record also from, we get also tissue from other areas. We always get association cortex. So we almost never get primary sensory areas for obvious reasons from the surgery. So the frontal areas, association areas, compare very much in terms of cell properties with the temporal association areas. So that's very comparable also in the morphology. So the complexity of pyramidal neurons is also very large there. You do see differences in complexity in pyramidal neurons when you compare primary sensory areas, but that's then from post-mortem studies. So in our studies, we don't have enough data to really compare primary sensory areas. So then, so why layer 2, 3? So that's a good point. We did in that study where I showed the strong correlation between layer 2, 3 thickness and intelligence scores, IQ scores. We did observe that we also measured the other layers, of course, but the strong correlation was really for layer 2, 3. So why is layer 2, 3? So this, what you have to realize is that layer 2, 3 pyramidal neurons in humans, especially the big ones, are much better compared with layer 5 neurons in rodents. So there's a sort of change of the big neurons in the human cortex that have moved supragranular. So instead of infragranular, like in the rodents, where the big neurons are really below layer 4. In humans, the big neurons can be above layer 4. And also, neurons, especially when you look at the transcriptomics of the neurons, in the human cortex, neurons don't respect boundaries, layer boundaries too much. They cross layer boundaries much more than in the rodent cortex. So I think that layer 2, 3. I think there's a lot of projections also going out of the cortex from layer 2, 3 in humans, but definitely horizontal connection and processing within the cortex is, of course, relies a lot on layer 2, 3. So that's also my best guess. It would be great to test that somehow, but I have no good experiment in mind yet. And then the third question was IQ score. So IQ score is, of course, a very simplistic measurement. So it's very practical for our type of work because getting this data out of the hospital is really difficult. So we're in the science faculty, communicating with clinicians. So you really need to find the right clinician to talk to. And then getting all that additional information from the patients, from the clinics, is really hard. We're really happy if we can just get one number per patient. That's already a lot. But we did in that study with the different layers, we also looked at different aspects of the IQ test. So you have fluent and you have crystallized intelligence, you have verbal, you have non-verbal. So we looked at those different parameters. They all correlate with the thickness. The verbal much better than the non-verbal ones. And then, of course, the IQ is, of course, a very simplistic view of the human brain. I mean, of course, humans are much more than IQ. So humans also have on the right side of for instance, on the right side of the brain, specializations that you don't see reflected necessarily in verbal aspects of the IQ test. So I'm sure that when you would do the right test for that, you would find a similar correlation with thickness and that particular cognitive skill that's represented by that part of the brain. But we're very limited to what the psychologists in the hospital do with the patients.
Hi, thanks for the nice talk. So I had two questions. So the first one is, so we've seen how the individual properties, so properties of the individual neuron, let's say, reflect into the human capabilities, human intelligence, but how does these properties project into the collective structure, let's say, of the network? For instance, can you say something about the way the neural network itself is structured in humans with respect to other species, or whether the structures or the connectivity, for instance, is transversal on different species, so it doesn't tell you much. And then the second question was, so we've seen the genetic reasons for these properties to develop. How instead these properties are related to nutrition, for instance, so to the energies available from the environment.
Okay, so the first question is how does this shape microcircuits in the human cortex or circuits and compared to other species? So I showed you only a couple of morphologies here and there. Then you really look at brain-wide studies that are being done now with, for instance, transcriptomics and coming from the Brain Initiative. What you really see is that it's not so much that there's really neurons, unique neurons in the human brain. No, it's really it's not a matter of, let's say, there's a different kind of neurons. It's much more there's much more specializations of the existing types of neurons that you find in all the other primates as well. But the relative contribution of these different kinds and their exaggerations of some of their properties is much larger in the human cortex. So for instance, this layer 2-3 expansion has resulted in a much larger amount of these big pyramidal neurons that you see much less in the other primate species, and even in mice you hardly see them. The same holds true for the interneurons. So the interneuron types, so there's a similar, let's say, diversity across the primate species. But the types, the representation of the types is different in the different species, also in the different cortical areas that you look. So, for instance, the double bouquet cell is a famous one. You see them mainly in primates, not so much in rodents. But in humans, they are very pronounced. So they have their axonal fields like in a columnar fashion, looks like they wrap around dendrites of these big pyramidal neurons. So this is a typical microcircuit structure that you don't see in rodents, that you see in other primates, but we think that it has to do with very fast interactions between these GABAergic double bouquet cells and the big pyramidal neurons, but it's all subject to study. So the second question was, let me see, so
Whether nutrition plays a role.
oh, nutrition, yeah, exactly. So energy supply of neurons. Yeah, that's a very interesting point. So what people observe in other studies is that the interaction between neurons and glia cells in human cortex is very different from, for instance, rodent cortex. What you see is, first of all, when you look at the normal protoplasmic glial cells, you see that they're much larger in human cortex. They cover much more territory than in rodents, for instance. But also when you look at oligodendrocyte precursor cells, OPCs, they have cell body to cell body interactions with all the large pyramidal neurons in the human cortex. In mice, you hardly see those direct interactions between OPCs and pyramidal neurons. So they think that these transfer directly resources to the neurons. So I think there's still a lot to be discovered. I mean, we did not do functional experiments on that yet, but I think in that corner there's a lot to be discovered in terms of how large pyramidal neurons in the human cortex maintain their functionality when they're challenged.
Yeah. So I had a question. Thank you very much for a fascinating talk. So maybe slightly misaligned question, because I mean, we tend to think we're great. I mean, some of us more than others, of course.
Sure.
But say an alien came 30,000 years ago, right? Would have found just an ape like other species, but biologically, as far as I understand, 30,000 years ago, humans were fully evolved, so there haven't been radical changes at the evolutionary time scale, at least.
Right.
But we couldn't do any of the things we can do at
Yeah.
So how much of biology is intelligence is actually biological? How much is cultural?
Yeah, so great. Thanks for this tangential question. It's so I can only speculate, of course. So if you allow me to speculate, then so these, I think that definitely the biology that we observe in the human circuits is not unique to humans at all. So I think that, for instance, this fast exponential timing to, let's say, 1,000 hertz is not unique in biology. So flies, the fly visual system, all the neurons can do that probably even faster than but the critical difference is that humans have that in an association cortex, not in their visual system, well, maybe also in the visual system, but not like the fly. But these fast properties, the humans have that in their association areas. So, of course, they had that biology probably also 30,000 years ago, you're right. But but it is, I think it's an enabling property. So it's not a defining property in itself, but an enabling property. But you need to make use of or challenge that enabling property. So, of course, when you look at our culture and compare it to 30,000 years ago, what's the big difference? I think we over-enrich our kids as much as possible. So when they're 1, we already want them to start reading. When they're 2, they need to speak fluent sentences. Well, of course, we try to push them at least. And when they're 4, they go to school, and they're in school until they're 24 or so. So we train and train and train and train, and of course that's when you start to see the expression of these properties are used to its full extent, or maybe it can be even more. But I mean, so that's what I think is the big difference between 30,000 years ago and now, is that the enabling biology was there already 30,000 years ago, but to really harvest that, you really need to culturally and behaviorally challenge that property. My speculation, I don't know.
Yeah, a fantastic talk. The question about the second part.
Mm-hmm.
So I was wondering about the oscillations.
Yeah.
They are often said to be related to movement, right?
Yeah.
Right, and so maybe to vision or to perception. But that is completely different in mice compared to humans, right? Also, the frequencies there are different.
Yeah.
So what's the reason for the brain to try to equalize these frequencies, or the center frequencies for the different versions?
Yeah, that's a good point. So, of course, again, I don't know what the brain, for what kind of reasons the brain has to synchronize all this at the same frequencies. I think that it is related to really the type of information that's being processed. So the information processing properties, with using biological substrates such as neurons, you require to have these kind of, let's say, synchronies between neurons going. And so it needs to be in that realm, in that ballpark, and that it is, I mean, of course, I think there's also kind of a little bit of an artificiality connected to it, because, of course, we define the gamma band as being between 30 and whatever, 80 or so. So that it's going to be between 30 and 80, that is already predefined, but it is the peak is also pretty much aligned, pretty well aligned. And of course also let's say the gross functional correlates, like being awake, having eyes open, and processing sensory information, that will generate gamma frequencies and beta frequencies. And that happens in mice, and that happens in humans too. But of course, when you start to zoom in onto the details, then you can find very big differences. So I think, again, it is the, let's say, you look into the enabling or the basic properties of how a biological tissue, like the brain, a mammalian brain, how that comes into an information processing mode. You need these ranges of frequencies, and then you can do a lot. And then, of course, on top of that, all those size and number of neurons and molecular adaptations, cellular adaptations, then you really get the specializations of the different species out.
Very beautiful talk. Thank you so much. I have a couple of related curiosity. The first one is if you found any main difference in the substructural architecture of spines in humans versus mice, and related to this, if did you quantify somehow the degree of local compartmental processing of information in pyramids from the two different systems? Thank you.
Yeah, thank you very much. So very interesting, super fascinating questions, because that's what exactly, well, one of the things, the spines, our colleague in Madrid, Javier de Philippe, spent a lot of time, and his team, of course, a lot of time reconstructing spines in humans and other animals. The density of spines it is a little bit higher in human parameters, but it depends a little bit on where on the dendrite you look. It is very comparable in terms of densities, but then the spines can be a bit longer. So there are details that are different. We looked at spines only in aging, but in a very, let's say, rudimentary fashion. And we do see that in aging, the spines are lost in human cortex. But at the spine level, I don't think there's huge differences between the species. In terms of compartmentalized processing and information processing, so that's definitely what we want to know. So these big dendrites, we've seen them in hippocampus CA1, huge dendrites in human hippocampus, huge dendrites in they must allow compartmentalized processing locally on the dendrite. So when Idan Segev made the models, he finds also that when you have these huge dendrites and these human dendrites of our data, they allow for many more different patterns of activation so that they can store much more information. We are looking at calcium signaling at the moment. We know that when you generate an action potential in the cell body, it travels much faster into the dendrite than in the mouse dendrites, but we don't know about the reverse. So one of the things we're doing is the glutamate engaging and looking at processing with calcium imaging in the dendrite. So this is an ongoing, we don't know the answers yet, but my guess is that there's much more local processing. Yes.
Yeah, so maybe I'm also venturing a bit into speculation. But I was wondering, I guess that to develop this kind of specialized neurons is very expensive for an organism. And so maybe the question is, if you use an evolutionary lens, do you have a way to, like, I don't know, I don't have a clever way to state this, but like to take the environment of the species, like a rat or a human, and try to infer what kind of neurons you will find? Like if you study neighboring species, for instance, monkeys.
Yeah. So one famous example are these von Ekenermon neurons. I don't know if you've heard of those. These are really large excitatory cells that are present, a lot of present in the human brain, especially in the insular cortex and also a little bit in temporal cortex, but in very specialized cortical regions that are thought to be related to social interactions. So of course one of the evolutionary advantages that humans have developed is acting socially together against whatever the prey or whatever, to collaborate and teach each other how to do stuff. So there are people that think that kind of specialized neurons and specialized circuits are really supporting that function. Of course, what came first, I don't know, chicken or egg, whether these neurons were there and then we were social, or whether we were social and then these neurons came, I don't know, but at least I do think that these kind of evolutionary pressures and specializations of organisms into certain niches of their environment and how to deal with the environment results in brain circuits being differently developed. And especially, I mean, of course, in humans, the huge increase in the association areas is probably a good example of that.
Thank you very much.
Thank you. My pleasure.